Hi there. Well, I will try not to keep you too long from your dinner. But so thank you for this opportunity to talk to you about some of the work that we've been doing at the Institute for Genomic Medicine at Nationwide Children's Hospital in Columbus, Ohio. So I'm fortunate enough to direct the Computational Genomics Group, which is a group of talented computational biologists and bioinformatics scientists and software developers. And a few years ago, we had a retreat to try to work out what was the mission of our group, what was the purpose of this group in a children's hospital in an Institute for Genomic Medicine. And we came up with this as our vision statement: solving the puzzle of genetic disease using bits and bytes. So if you're here today to hear me give you the answer to that puzzle, you're not going to hear that, but what you are going to hear is an invitation for you to join us as we try to solve this puzzle. So how we've been approaching different genomic diseases or different genetic conditions is through what we call translational protocols. And in the Institute for Genomic Medicine, or IGM, we currently have about 10 translational protocols that we've launched over the years. What each of these protocols does is it attempts to connect clinicians, researchers, and the bioinformatics scientists and computational biologists together to target a particular disease. And what you can see in that bar chart on the right is growth of these protocols over the last 5 years. And today I'm only going to tell you about two of them, the rare disease protocol and the cancer protocol. So these beautiful kiddos are some of the nearly over 7,000 different types, faces of kids with nearly over 7,000 different types of rare disease. Now, just because a disease is labeled rare, it doesn't mean that it's insignificant. It's estimated that as many as 1 in 10 Americans are affected by a rare disease. And if you take my own immediate family, there's 3 of us that are impacted by 3 different rare diseases. One of the statistics that we've been trying to really focus on this slide is that that figure down there of 8 years on average, but for which a child receives a diagnosis. Unfortunately, with the advent of next generation sequencing technology and genomic approaches, that time is coming down and we're able to give more and more kids answers quickly. So in the Genomics of Rare Disease protocol, as of yesterday, we'd enrolled 220 patients. And what these patients, to qualify for this study, they have to have undergone the current standard of care, which is exome sequencing and all of the other appropriate clinical testing. And they've done that and it's been negative. So what we're able to do is enroll them into our study, enroll their unaffected family members, or if there's an affected family member, we'll enroll those individuals. And we perform genome sequencing along with RNA-seq on the RNA, so on the blood RNA from the blood RNA, or if we have an affected tissue, we'll do RNA-seq on the tissue in an attempt to find out what's going on with these kids. Now you can see on the pie chart there shows some of our results so far. So for the 184 cases that we've completed, 53 of them we've been able to solve. So this, by solved, we've been able to identify what the pathogenic variant was that was causing the kid's disease and provide that answer back to the clinician and the family. Many times this has required collaborating with biologists and performing functional validation to prove that that is the causal variant. And then that red part on the left is our negative cases. So still about just over a third of cases we have no result. We haven't been able to find an answer for those kids. But that bottom piece of the pie, that chart, is what we call our candidate gene piece. So for about 1/3 of the cases, we've been able to find a gene, but for that gene, we've not been able to meet the burden of proof that it's actually pathogenic. So work is ongoing to demonstrate that the variant that we've found is relevant for that disease. Now, one of the ways that has been tremendously helpful in moving this program forward is in data sharing. So for many many groups doing Mendelian disease research, there's a number of resources out there in the community to share information. And we've used a resource called GeneMatcher, which is part of the Matchmaker Exchange Consortium. And what this platform does is it allows geneticists and researchers to share information about their patients, the gene that they're interested in, and the particular variants. And then when you submit a gene, everyone else that's interested in that gene is sent a notification message saying there's been a new submission, and then you can compare results and what your phenotypes were in your patient. So to date, we've submitted 82 genes to this resource, and for 10 of them, we've been able to publish, describing new genes. And for 15, we currently have manuscripts in progress that are part of larger cohort studies where our patient is being included in another group study. So the second half of the talk, I want to tell you about our cancer protocol. So brain cancer is currently the leading cause of disease-related death in children. Over 90% of kids that are diagnosed with a brain tumor will eventually succumb to their disease. So what we set out to do in January of 2018 was to start what we call the IGM Comprehensive Cancer Genomics Protocol. And what this protocol has done is we've focused on kids with CNS tumors and rare tumors or relapse disease, and then perform comprehensive molecular profiling in terms of exome sequencing, RNA sequencing, and epigenomic analysis by looking at the methylome. For any of these patients that have enrolled in this study, if we've had a research finding that we've thought was relevant, that the team has looked at and thought was relevant to that patient's disease, they've undergone clinical confirmation in our clinical lab so that those results can be returned to the clinician and the family and entered into the medical record. So to date, we've enrolled about nearly 450 patients on this protocol. And the figure there that I really want to point out is that for 90% of the patients that we've enrolled, we've found a medically meaningful finding in the genomic analysis. And by medically meaningful, I mean in terms of we've found a variant that's either diagnostic, prognostic, or therapeutically relevant in terms of perhaps it's an indicator of a targetable gene or a targetable mutation for a novel therapy. So with this data set, we've been very committed to sharing all the data that we generate, all of the raw data and our results, and we've been routinely uploading the data to dbGaP. So if you're interested and want to get involved in looking at this data, it's available. If you just, I think if you Google dbGaP and Nationwide Children's Hospital, it's the top hit. So if you're trying to write that number down, don't worry. So as a result of the work that we've done on our cancer genomics protocol, we were recently awarded a contract from the National Cancer Institute called the Molecular Characterization Initiative, or MCI. And what this initiative is, it's a program that's we're working in collaboration with the Children's Oncology Group, enrolling patients from across the nation to receive similar molecular profiling that I described to you in the previous slide. We receive samples from biorepositories, and we've set up a system by which we receive electronic data in terms of the patient's case manifest. That's all incorporated into our laboratory information management system. Then the lab performs the molecular analysis. For our bioinformatics processing, we've fully automated everything using a platform in the our genomics platform in the Amazon cloud, and then all results are analyzed, interpreted. And what's unique about this is the patients receive a clinical report. This is all done under regulated standards that are CAP clear and New York State approved. Finally, all data is delivered through a portal back to the clinician, and also it's delivered to a public repository for everyone to be able to access. So these are the assays that we're doing. We're looking at the genome in terms of an exome analysis, where it looks at the patient's tumor sample and compares that tumor genome to the germline genome from their blood, from a normal comparator. Those exome sequencing is done at a very high depth of 250x, which allows us to detect variants that are present at a low variant allele frequency. We also have spiked in copy number probes into the exome that allows us to report out copy number variants and LOH. For the transcriptome, we're using a very sensitive assay that looks specifically for known cancer fusions from a company called Archer. And then finally, for the epigenome, we're using the Illumina EPIC 850K methyl array and using the DKFZ classifier for disease classification. All of these assays are then reported out clinically within 14 days of receiving the analytes. The last part I want to talk about in this protocol is the actual raw data is then being shared. And what we've had to do, because this has all been done clinically, clinical regulations require that every sample and every file that's generated contains 2 patient identifiers. Now, for data sharing, that's obviously a problem because we don't want to make any of this information public in terms of the identity of the individuals that have enrolled in the study. So we've actually developed methodology to completely scrub those identifiers and replace them with a unique sample identifier and deliver the data to the CCDI in an Amazon S3 bucket. Now, we've had to scale this up very rapidly because the clinicians got very excited about this new protocol, wanting to enroll their patients. We're now up to about 10 cases a day. We were contractually committed to deliver the data within 90 days of a case being signed out. But what we did is we developed a system that automated data delivery so that within minutes of a clinical director signing out the case, all of the data is sent over to the NIH CCDI group. Who is CCDI? Well, they are the Childhood Cancer Data Initiative. They're the group that's actually responsible for sharing all of this data with the community. So what they receive from us is the raw data in terms of CRAMs, VCFs, the EPIC array IDAT files, and all of the clinical reports that have been de-identified. They also work with the Children's Oncology Group to gather all of the clinical data. So it's not just the genomic data, you have the clinical data that's available. And finally, they release all of that data to dbGaP, where it's available for general research use. Now, I spoke to the folks at CCDI last week to ask them the question of when will this be available? And they said they've had lots of inquiries about when it will be available. They're working really hard to get this up. They're anticipating September of this year. So far, I think we've enrolled about 130 patients have gone through the sequencing process. So come September, there'll be over 200 to 300 cases that are available for the community to look at. So I'll end on a story about why we're doing this. If you remember that statistic, 90% of kids that are diagnosed with a brain tumor will succumb to their disease. Well, this is Jacob's story. He presented at Nationwide Children's back in 2013 with a rare brain tumor that was removed surgically in January of 2014. At the time, they performed genetic testing using a commercially available panel test, but didn't find anything of relevance for Jacob's disease. He underwent proton therapy and chemotherapy. 4 years later, in 2018, his disease had reoccurred. At this point, we enrolled him into our study. He was one of our very first patients. We performed the genomic profiling after his tumor was resected. We took the tumor DNA and RNA, performed the genomic profiling, and we found that he had a druggable mutation in the gene called PDGFR beta. And what this gene is, it's targetable with a therapy called dasatinib. He was started on that therapy, and then this is the results from last month. So this is from June last month, and the image is labeled NED. NED is good. It stands for no evidence of disease. So if you have a brain tumor, that's the best result that you can get. So with that, I do want to take a second to thank all of the people in my group, in the computational genomics group, our IGM clinical lab, NCI, Children's Oncology Group, and the Childhood Cancer Data Initiative, and of course the patients and their families. And if you have a chance, we have a number of our trainees and postdocs and staff are here presenting. At the bottom 4 are talks, the top 4 are posters. They'll be happy to tell you more about the work that we're doing in IGM. Thank you. Sure.
Because it's rare diseases, how do you ensure privacy? Because the probability of having a disease is so rare that it's possible to be real.
Yeah. So the question was, how do we ensure privacy for the individual, given it's a rare disease? So that's something that when we consent all of our patients, we do say that we'll be sharing their data in controlled access databases. So that's one way, is that the only way to get this data is you have to go through the process of being approved for controlled access. You also have to agree to not attempt to identify the subject from that data. But you're absolutely right. Some of these genetic variations are so rare they're probably the only person on the planet that has that particular variant. The only way to find them is if they're in a database that would have a match to that. Yeah, it's something that we have to consider as well. Great question. Sure.